Embarking on a microscopic journey into the intricate world of parasitic nematodes, one encounters the rhabditiform larvae of strongyloids, diminutive organisms that epitomize both the elegance and complexity of the natural world. This is what we unravel while performing direct saline wet mount of stool from the patient in medical ICU. A case of strong helminthic parasitic infection where we detected many larvae of Strongyloides stercoralis. Under the microscope, the rhabditiform larvae of strongyloids appear as slender, elongated organisms with a characteristic cylindrical body. They measure approximately 250 to 300 micrometers in length and exhibit a translucency that allows for the visualization of internal structures. This transparency, coupled with their simple yet elegant anatomical features, makes them a subject of fascination and study within the realms of parasitology and microbiology. One of the most striking features of the rhabditiform larvae is their well-defined cuticle, a protective outer layer that provides structural integrity and defense against environmental stressors. This cuticle, adorned with fine striations, lends the larvae a sleek and streamlined appearance, facilitating their movement through soil particles and facilitating their survival outside of a host organism. As they navigate through their habitat, rhabditiform larvae exhibit a characteristic mode of locomotion, known as rhabditiform movement. This mode of movement involves a sinuous, wriggling motion propelled by the coordinated contraction and relaxation of their longitudinal muscles. This efficient form of locomotion allows them to traverse the intricate maze of soil particles in search of nourishment and suitable hosts. Internally, the rhabditiform larvae of strongyloids possess a rudimentary digestive system consisting of a simple intestine that extends longitudinally along their body. This intestine, lined with specialized cells responsible for nutrient absorption, enables the larvae to extract essential sustenance from their environment, ensuring their survival during the larval stage. In addition to their digestive system, rhabditiform larvae also harbor reproductive organs in the form of undeveloped gonads, which will undergo further maturation and differentiation in subsequent stages of their life cycle. The diagnostic importance of strongyloids larvae lies in their ability to serve as key indicators of infection in both humans and animals. Detecting these larvae in clinical specimens or fecal samples is crucial for accurate diagnosis and timely intervention. One significant aspect of strongyloids larvae in diagnosis is their distinct morphology, which allows for reliable identification under microscopic examination. By observing the characteristic features such as the slender, elongated body and the presence of a buccal cavity with stylets, healthcare professionals can differentiate strongyloids larvae from other parasitic organisms with similar life stages.